about his area of expertise. So with great pleasure, I hand over to Ken to talk about, it was a bit of a scary title at first, the hidden cancer, but the need for improved treatments and outcomes. Thank you, Ken.
Thanks very much, Gail, and thanks very much for coming out on a really damp and horrible night, and you're very welcome. I'm going to be moving around, and the talk is going to be called The Hidden Cancer, the Need for Improved Treatments and Outcomes. I'm based in Belfast, at the Queen's University Belfast, in the Patrick G. Johnson Centre for Cancer Research. Only got called that about 3 weeks ago from the previous name for the Centre for Cancer Research and Cell Biology, after Paddy Johnson, who was our Vice Chancellor and the first Director, died very suddenly 2 or 3 years ago. So he was an amazing person for cancer research. So I just thought we'll show you where we are. This hopefully is going to play. We're going to give this talk, give you building up about what we do and where we're going to go. As we're zooming across the Medical Biology Centre, Lisbon Road, into this building here, which is where I work, was called the Cancer Research, and it's now Patrick G. Johnson Centre for Cancer Research. Just to give you some sort of flavour for what happens in the labs, a very short video, just seminars, foyer, cell biology, back into the tissue culture. Just as a very quick overview about where I come from, because maybe people don't know Belfast particularly well. So, I will give you this health warning now, if this works. Yes, there will be questions ahead. I'm going to involve you, I'm going to ask you questions, and there's actually going to be certain sections that will be audience participation. So, please, if you're not happy with that, please say no. So first of all, I want you to put your arm up if you've ever heard of it. Waldenstone macroglobulinemia. Good. I was hoping you might do that. I'll be worried if you hadn't. Systemic mastocytosis. We'll just assume Ben's put his hand up. Mast cell sarcoma. Okay. Getting there. Myeloma. A few more. Lymphoma. And leukemia. Hopefully everybody's probably heard of that. Okay, so all of these are actually blood cancers. There's actually 137 different types of blood cancer. So that's part of the thing is why it's called a hidden cancer. And the thing, some of the names around the outside here, the names around here, none of them actually have the word cancer in their name and their title, which is standing beside the radiator, it's really warm. So that is part of the problem, in that when people don't know and don't appreciate about blood cancers, is that there's so many various types and so many different aspects to that disease, really. So to put it into a context, blood cancer is actually, here's the big five, I suppose: breast cancer, which you probably might have predicted, prostate, lung, bowel, and surprisingly, blood cancer as a group comes in at number 5. It's the 5th incidence of cancers, but nobody really thinks about and talks about that, because you all talk about, or people who relate to, specific type of lymphoma, myeloma, or leukemia. What is equally as scary is that this is the proportion of people who might die with that particular type of disease. And you can see that a high proportion of people with blood cancers, it's the 3rd incidence for deaths. So even though it's the 5th incidence, it actually kills more people from breast cancer or prostate cancer does, which has a higher incidence. So that's again why we consider it a hidden cancer. And here, take this 50,000 people, this is the accumulation of the lifetime incidence over, and you can see as the ages go up to 50, 60, 70, you can see how more people are going to be affected by a blood cancer disease. So there's the people affected, and there's a group, so 1 person in every 19 is going to be affected by a blood cancer during their lifetime. So that's actually quite a a surprising fact when people think about it, which is one of the reasons why it's called the hidden cancer, because people don't know too much about what goes on and what that type is. And it's called the hidden cancer because this report came out from the All-Party Parliamentary Group in Westminster, where they're trying to improve blood cancer awareness and blood cancer treatments because there's also a lack of therapies for some of the types of these cancers. So, I said there's 3 main types. About 23% of all of blood cancers are leukemia. Almost 60% of blood cancers will be involved in lymphoma, and lymphoma itself has about 59 different subtypes of lymphoma. About 12% of cases are myeloma, which affect the bones, and 6% for all the other 137 different types. So, and the incidence increases with age. Well, it's not unusual to most cancers do. Cancer is a disease of aging, so you find that with all of these ones, the cancer incidence will increase. Different diseases have different rates. But if you ask a lot of people in the street who might get leukemia, it's always children. They're the most group, but it's not. Only about 1% of cases would be in patients under 15. It is a disease of young people. If people do get cancer, it tends to be a leukemia or a brain tumour, but that's basically the 2 different types. 75% of all blood cancers occur in people over 60, and if you put it into the Isle of Man context, about 1 person every week is diagnosed in the Isle of Man with a blood cancer. So what is blood cancer? Well, it comes from this thing called the bones. Your blood is made in the bones. How much of your body do you think is covered in blood? Out of 100%, what percentage do you think represents blood? Anybody going to give me an answer? Or are you going to be like my undergrads and just sit quiet?
63.
63. So, slightly lower than that, but so 5%. 55%. No, you're 12. A bit higher than that. Higher. Well, nearly 25% of your whole body is blood. So what we're showing here is the marrow out of the blood, out of the bone, put into culture, and you can see how fast it's growing. So basically, you've got...
weight or volume?
Sorry?
Is that weight or volume?
Both really. So it's a lot of your body is blood. So if we just go back, I was talking over that one. So if you take a piece of bone, in this case this comes from a mouse, it's not a human bone, and squeeze the bone marrow out into a petri dish that has medium growing, so you can grow that. So you can see there's the marrow, line it out, and it's red because it's full of red blood cells as well. So all your blood comes from the bone marrow. It comes and grows at a very fast rate, because if you think about how much blood you're making at any one particular time, and you're making, how many cells in your body? That's the next question we're going to ask. How many cells are actually in your body? Trillions, that's a good answer actually. It's 100 million cells. So if you've got blood there, that's 25 million cells to be made. And the lifespan of some of your red cells is about 120 days. So you have to replace all that. So, and like anything, it's a balance between life and death. Cells will grow up, mature, do their job in the blood system for 120 days, and then they die off and get recycled. Otherwise, we'd all end up as a big red blobs, and that wouldn't be very pleasant. So there's the bone. Here, this is your bone. And in here, there's this little cell here. One cell amongst all these bones here. And those are called stem cells. And those stem cells produce all the blood for all of your life. And there's another number I'm going to ask you for. How many stem cells do you think you might have to keep all your blood system going for the whole life. Nobody thinks. 1,000. So not many really, considering, but it's got to produce those 25 million cells all the time.
They don't die often.
Sorry?
They don't die off the stem cells.
No. Well, they may do, but they're not all active at once either. So you might have some who are sleeping and other ones who are more active. And so, but they gradually die off, I suppose, or get worn out during your lifespan, which is why people end up with different diseases as they get older. So that hematopoietic stem cell, this thing here, produces all the blood from here. So these things over here are all the mature cells that you might find circulating in your normal blood. So there it is, there's a stem cell. We put it into a culture and it starts dividing. Interestingly, one of these stem cells would become a stem cell and the other ones would start to produce all these ones up here. Now if we put different ingredients into each of the different cultures, we can turn them into different cells. So if you look, they all started from that one stem cell, but they all start and look at different shapes. They're different sizes and shapes. They're clustering, some are growing faster, and you'll notice for this one here will start to make red blood cells and start to turn red. As you can see, that's making a hemoglobin, which you know because if you cut yourself, you get red. About 1 in 1,000 cells, every 1,000 cells are red cells. About 1 of those cells in 1,000 are white cells. These things here, the neutrophils, eosinophils and megakaryocytes. And they're the things that should be in your blood. So putting it in a different way, it's actually compulsory if you're talking about blood to show at least one of these pictures. Otherwise the blood police come down and tell me off. But the stem cell here, this is the thing that we just seen up here. There it is there. That's the stem cell. And down here is those things that we saw, the blood cells, the neutrophil, basophil and eosinophils. And all this occurs in the bone marrow, and it's only the mature cells that occur in the peripheral blood. So if you think about the stem cell as being sort of like a baby, this grows up, becomes maturing, goes to university, comes out and starts being a productive member of the society, which is what the blood cells do in the peripheral blood. So if something happens back here in the stem cell, DNA damage, something upsets the DNA in that stem cell, it's going to have an effect up here, but it will also manifest itself here because it will block the growth of those particular mature cells here. So basically, you're blocking adulthood and ending up with a whole load of delinquent teenager blood cells in your bone marrow. And that blocks it in some stage here. So it can block the differentiation cell death in some of the more maturing ones. But basically, it's blocking the growth of these cells here. So you're not having an active population of blood cells in the peripheral blood. So what that means is that you have all these different aspects, and where the effect happens, remember the defect happened up here, but if it's in this region that the thing becomes manifest, you get acute myeloid leukemia, you get acute lymphoblastic if it's over here in the lymphoid side, and so on. So depending on where the effect or the aberration happens, depends on where the disease occurs or what the outcome of it is. So because we haven't got time to talk about all 137, you'll be pleased to know, we're going to talk about 2 of them. And we're going to focus on this side, the myeloid side. We're going to talk about chronic myeloid leukemia and acute myeloid leukemia, and to give you a different compare and contrast about what the outcomes and treatments are and the complexity of the disease. So chronic myeloid leukemia, I'm going to call it CML because it's easier. So here's what a normal blood film looks like. These little brown spots are your red blood cells. So brown is red and white cells are blue. It's really confusing. So in this case, this is what your normal peripheral blood should look like. And in this case, this just gives you some numbers. This is about 5 to 11 times 10 to the 9 white cells in a litre. Now this is a chronic myeloid peripheral blood. I think you can all see, sorry, I'm on your white go, but you can see the number of excess. There's more cells here than there is in here. And the white cell count can go from 8 to 800. So probably 100 times more red white cells in your blood. The reason there is 100, these are more mature. These are actually look like normal adult blood cells. The difference, the problem is that they don't die. They're like they're supposed to. They just keep on accumulating in the blood. So, let's go back and we have to have a bit of a genetics lesson. Sorry about that. So I'm sure you've all heard about DNA. And DNA contains genes, and genes make proteins. The DNA is wrapped up into chromosomes. And so, I have my rubber tubing here. Gail didn't mention the rubber tubing, but
I was shy.
she's a bit worried about the rubber tubing. So I've got rubber tubing here. It's naturally tied itself up into things like that. So this rubber tubing, once I get it untied, is 2 meters long, each of these. Do anybody know why it might be 2 meters long? It's the length of the DNA in every cell of your body. So that's how much DNA is in there. So if you unwrap that, maybe I can ask you, Gail, and this gentleman here, to hold that end and hold that end. And if you just start twisting the rubber together, turning it around like that, yes, just turn it around. And so basically, what each of that DNA has to be packed up into each of the cells. So it does that by basically supercoiling it into chromosomes. So these are chromosomes, and they end up in the nucleus. So over here, we've got, that's the chromosomes, which we'll come to in a minute. So, okay, that's fine, let's just hopefully this is going to work. So if we now do that, and basically what it is now that you can see, because they've twisted it together, it's gone from 2 meters down to 70 centimeters. And so if I carried on doing that, that's exactly what happens in a chromosome. DNA is supercoiled. It's coiled, but it also has to have accessibility, because you've got to make proteins. Now, if you just assume that some of these red ones here are going to be genes you want, they've got to be unwrapped, so the DNA has to be allowed to be unwrapped. It's a bit like your house. If you want something in your kitchen, you have it on your work surface. Your kettle you use every day. So that's your active genes. The ones that you're going to use occasionally, you might have in the kitchen cupboard, and things that you want to store but you don't want to throw away, you put in the attic. And that's what happens on DNA as well. There's bits that you want to be expressed and have activity. There's bits like this black one that you may want to express only in certain parts of your body and at certain times under stress, and other bits that you didn't need because you needed them when you were growing up as an embryo, but you don't need now. Okay, so it's okay, I'll get them done later. So, this is what this chromosomes look like if you took them out of the cell. And here is a set of chromosomes here. You've got actually 46, I've only got 24 over here. So they go from 1 to 22, and you've got an X and a Y chromosome. Hands up, who's got a Y chromosome? Good, none of the female, only the males have. That's fine, that's good, that's a good test. And so they are then to pairs down here, and you have the big X and the little Y's down here, the bottom down here. This is actually a female chromosome spread, so there's 2 X's. So I'm not sure why the Y chromosome in men's is so small, but probably the females have got an explanation for that. So in the 1960s, these 2 people, Noel and Hungerford, found that this little chromosome here, what they call chromosome 22, or derivative of chromosome 22, this little fellow over here, the cute one, was even smaller, and they called it the Philadelphia chromosome, because they discovered it in Philadelphia. And so that was in the 1960s. It went on to show that here was this little chromosome here, the Philadelphia chromosome, had a break and had swapped material with chromosome 9 and causing a translocation. Now, I haven't got chromosome 9 and 22, but I've got chromosome 9 and 15 and 17. So I'm going to pick on two different people now. Giovanni, you can have number 15. Carol, you can have number 17. So what we're going to do is we're going to actually have a genetic abnormality occurring in this room here now. So we've got chromosome 17 over there, 17, chromosome 15. I'd like you to swap material to pull its head off, basically. Okay, swap that over. You can't see, so basically now you have two chromosomes. They haven't lost any material. They haven't gained any material. But what they have done is that they've swapped it around. So here you've got, well, it's 15, 17, it says that there. So that's quite good. So you've got, at this particular breakpoint here, at this junction here, you have the ability to make a unique protein. A protein that only occurs in that particular disease. So it's like taking two recipe books or two proteins, taking them apart and putting them together again. One could be a cake recipe and one could be a wall making concrete. So you can put them half together. That's not quite cheap because I've tasted cakes that taste like concrete before, but you know, you get the thing. So, whoops, what have I done? Oh, there. So there's chromosome 9, in this case, we'll forget about 15, 17, it's just, this is chromosome 9, this one here, chromosome 22 down here, and there's two little genes down here, one's called ABL, and one's called BCR. I don't really want you to remember all these genes because I'm going to throw other ones at you later. It's more to do with the concepts. So what happens there is those swap over, and you have that rearranged chromosome down here. And you have this fusion gene that only occurs in this particular disease, BCR ABL. So it's half a BCR and half of ABL gene. And that can be used, and that's the BCR ABL here. And what it does is it recruits in this substrate, which can be an activation. It gets this ATP activates that, and then it goes on to cause the leukemia. By using this drug here, Gleevec, it sits in this little pocket. So it stops ATP getting into the pocket, and therefore it stops, we go through it again, there's ATP, puts the phosphate group onto the substrate goes away and tells the DNA to make lots of different genes which block differentiation. Gleevec will sit in this pocket, will stop ATP, means that you can't put the substrate onto here, so you've stopped the leukemia growing. By one simple drug, you can stop that disease happening quite nicely because of this BCR-ABL. We can target this little pocket. And so when this came about, this Gleevec actually made the front cover of Time many years ago as being the wonder bullet, the magic bullet for this disease. And it's the war against cancer. So the idea was that you needed one pill to cure leukemia, which is true. There they are, somebody's taking them that. And it has had an amazing effect on this particular disease, chronic myeloid leukemia. Because it can get rid of this BCR-ABL, and if people are now 4 years without evidence of that protein being around, they can stop taking the drug. And they can stop taking the drug for the rest of their life. And the amazing thing is that before this drug came out, these are the different survival rates. Just look at how they join together. And now, people with chronic myeloid leukemia have the same life expectancy as the general population. So it's been an amazing turnaround. Just from one drug and one type of abnormality, which we understand. Okay, I've done it again. So we're going to switch now to acute myeloid leukemia. Now, if you remember, this is where acute myeloid leukemia is. It's the more immature cells. It's the more delinquent teenager type cells. And so there's the normal peripheral blood or normal bone marrow here, and here is the ugly ones. They all look the same. They're big, they're horrible. They've got granules in them, which are not very good because they could cause clotting and things like that. So It's a different type of disease to the one we just talked about. It affects a similar type of age population, still increases with age, more patients with AML than there is CML. The downside is that as you get older, or the patients diagnosed at different age groups, the survival rates are pretty poor. And you can see that somebody diagnosed over 75 has got a less than 1 year survival rate really compared to somebody under 15 with this particular disease. Not so good. Different from what we just said about CML. So, why don't we use the same type of approach as we just had for the CML? Take a patient, who's got BCR-ABL, give them this drug, imatinib or Gleevec, and it stops the growth. That sounds really brilliant, and that's what we should be able to do. But in this case, we might have different mutations because BCR-ABL isn't always in, very rarely in AML. So you need to look for patient A with mutation A, give them drug A, and maybe patient B with mutation B, give them drug B. Sounds all pretty straightforward, doesn't it? So what are mutations? So let's take a sequence. This is the alphabet of DNA, made up of A, C, G, and T. So if we have a sequence like this, and we mutate it, and substitute C for T, it can give you a protein that's missense. It's a protein that's still being made by the body, but it actually isn't very good, or it's doing the wrong thing. Either too much or not the right stuff. You could have an insertion. So you've got the same sequence either side of the red box, but now you've got an extra bit of protein added in, and that extra bit can cause you nonsense. It can actually make the whole protein not function at all, let alone function inadequately, or it could actually just block the protein completely so you don't have that present at all. And the same thing is if you have a deletion, you can have a nonsense or a truncated protein. So there's lots of different mutations and different ways of getting it. So if you talk back to the CML, how many genes did we have affected? Well, how many abnormalities? We have one, BCR-ABL. And BCR-ABL does occur in AML, and that's when it was discovered, 1985-ish. But if we now look to see how many mutations occur in AML, quite a few. And some of them are translocations. All the ones in boxes are like the BCR-ABL, the one we just done in our experiment. They are translocations, the rest are mutations. And you can see translocations we're discovering earlier, and more recently there have been more abnormalities and mutations. So in fact, when we look at it, and this data here was from a few years ago from a study, you can see that each of these columns represents a different gene. And there's actually 77 different mutations that can occur in this one particular type of disease. So that makes it slightly more difficult, for you may need 77 different drugs. So at the moment, we don't have that, so 77 different drugs for targeting different mutations. So how do the mutations occur? Well, they occur by DNA damage. DNA damage can come around by diet, infections, radiation, chemicals, just basically life. And so the DNA can get damaged, and you have, at the moment, even though you're sitting quite quietly, you're making 2 million red cells every second, which is pretty impressive, but you're also repairing around about 500,000 damages in your DNA. So you're actually quite effective at repairing DNA damage. So Dan, the DNA damage man, comes along and repairs that quite effectively in all cases. And why is that important? Well, because if a cell has too much damage, maybe it has a lot of radiation or it's something like that, the cell will die. It will recognise that it's too damaged to be repaired and die. If it can be repaired, you take it along and the cell survives. So the damage back to the garage, somebody repairs or changes your tyre, you can carry on driving it. If you have a major accident, you scrap the car. But if you have too much damage or inefficient DNA repair, the cell will survive but with the DNA damage. And if it's the right cell at the right time, like the stem cell, that it means that there's the start of a potential cancer. So in actual fact, even though any cancer is quite rare, it's rare in the biological terms because you've got 100 million cells, they're being replaced on average every 100 days, and you're all living to a good old age, but only one of them needs to be damaged to produce this cancer cell. So in biological times, having a cancer is extremely rare. But once it happens, it's pretty binary because you've either got it or you haven't. It doesn't make you feel any better, but it's a rare thing that happens, but it's still like that. So some of the mutations are effective in DNA damage. And what I don't want you to go away is go back and home and have a discussion over your tea on the relative merits of SF3B1 versus NPM1. But what I would like you to remember is if it's like when you take your car to the garage and you say, how bad is it? And the mechanic goes, oh, and you know it's going to be bad. But anything with a P53 in it, I give you permission to do that whenever you hear of anything with a P53. And you can rehearse that later if you like, going, that's bad. So a lot of these things here are involved on different constructs, and these are all involved on DNA damage repair. And there is also something that we were actually researching in Belfast. And just as an example, I want you to look at STAG2. STAG2 is involved on chromosome structure. This stuff we had a look at earlier, which I've now got to unwrap because somebody wrapped it all up for me. So I'm going to take one of these. And some cable ties. Gail was very worried when I said I got cable ties. Okay, so who wants to help me to construct some chromosome damage? Would you like to come up, sir? So STAG2 holds chromosome in the structure. So I'm going to put these two together, and can you put the chromosome? You are now acting as a cohesion molecule, right? So you put that together, loop them together. Now I want to loop the black ones together. Can you do that again? Just through there, No, through there, sorry. That one through there. Yeah.
Okay.
And the last one is a delinquent stag, so I'm going to put that one like that, so you can put it through like that. So this is why we need a Y chromosome.
Why?
Y chromosome. Okay, so, not quite better. So we have a structure here that's being held into place by the STAG mutations. The STAG cohesion molecules. There it is there. So the cable ties are this complex here. Here. And it's locked into place by this protein called STAG2. Occasionally you get STAG1. STAG1 sits at the end of the chromosome, so that's the STAG1. These two are STAG2s. So this holds it into place because this is very important, because now it allows access to these red parts, these proteins. They come to contact so they can get expressed. The black ones are all locked away, so they're not so good. You don't need the black ones. So if you have a mutation, I'm going to put that one down there. Pick up my other tube. So here it is, now that's locked the DNA into place. So if you have a STAG2 mutation, you don't have STAG2 because it's on the X chromosome. And you've only got one copy, even if you're a male, because the other copy is switched off by chemical inactivation. So you don't have any STAG2 to hold that. So, Gail,
Yes.
try and replicate that with one molecule, with one cable tie. Basically, you can't.
Okay.
You
I was
can't.
going to give it a go. But
You
I
can
can't.
give it a go, but
I'll give it a try.
you can't because you need 3 cable ties to put that into place. The only one you've got is this one here.
Yeah.
So basically, you've tied it together at the end. Up the chromatin back. See? So you've tied this together here, but you've got floppy chromatin. STAG2's mutated, so you've got this open, it lets it go. So you now have the opportunity to express these black genes. And they're the ones that were switched off in the normal system. So you have abnormal expression of genes that are not just the mutated ones, so you've got a whole construct of things that's going wrong. So, if we look at 200 patients, and you remember from the BCR-ABL and the CML that this gene, a gene, was specific across all of them. And here you can see this is 200 patients going down in columns, genes are going across in rows, and these are translocations, and you can see that because you can just see the blocks, they're all exclusive. You don't get one without the other. So some patients would have this one, some would have this one, some would have that one. So again, you think that's quite good. They're only going to have one particular type of mutation. But you can see, if you go down the line, how many different bars there are in each particular patient. So patients have more than one mutation. So again, that's a difference to the chronic myeloid leukemia. So if you just took 32 genes from 200 patients, you've got 125 different combinations of mutations in genes. So just look at this. We take this one here, NPM1, quite a snazzy little title. Half of them have this other gene, DMT3A. So some of them have got just one mutation, some have got two, some of them have got three, some of them have got two, some have only got one. And there's those ones here. So you can build up all this different complexity from this. So if you had a drug that targeted NPM1, that'd be really useful, but it may not work in the presence of this mutation here, or it may not work if these mutations are present plus NPM1. Or it may only work if this is NPM1 plus these mutations here. So you don't know what concept and what drugs there are. And there's 78 different mutation complexes just by using this particular number here. And that's fine, so it's a moving target perhaps. Here's a patient, and again, don't worry about the names, just look at the colours. This is a patient here at diagnosis, and you can see that it's got 1, 2, 3 different types of clones. They've got this gene, this one in common, different ones here, so there's a different set of genes. So you treat the patient at diagnosis and come back and look at the disease later, and you can see how the disease has changed. What you have at 1 year later bears no resemblance to what it was at diagnosis. So even though you may be treating patients and have a drug against IDH1, which there is, it actually disappears quite quickly with that therapy, but allows other mutations to be developed. So the disease evolves constantly, which again makes it more difficult for therapies. And here's a whole group of other ones. Again, don't worry about the names, just look at the colours. This patient didn't change at all, actually. Here you can see ones that do change quite rapidly, and some things go away and come back again. And so it's going to be a different way of treating patients, but you need to treat all the mutations at all the times and make sure you know which ones you're changing to. It's going to be not the same therapy all the time, but a changing therapy. So, just some of these mutations that I talked about today can be found in healthy individuals. And about 1% of the population have these mutations, and they increase in frequency. What do you think they may lead to? Remember, these are blood cancer related mutations. What do you think they may actually come out and give you? Only about less than 1% of people with those mutations, so 1% of 1%, might go on to develop a blood cancer. 40% will have it's an increase of 40% for cardiovascular disease. So, interestingly, by having a blood cancer mutation, but just the one, not anymore, but just one, could lead to an increase in heart attacks and heart rates. So it's a very complicated system we're looking at. That's potentially because you're increasing shape and size of the white cells, even though they look potentially normal. So, just what can we do? So I want to tell you about our initiative we're doing. It's called MyBlock. It's Myeloid Blood Cancer Initiative, which we hope will improve patient diagnosis and particularly improve therapeutic options with myeloid blood cancers. And this is just a paradigm, a way that we can do this and then move on to other types of blood cancers and cancers. So it's basically adopting a personalised medicine which we consider to improve patient outcomes. So what it involves is taking a patient sample, either at diagnosis, relapse or second relapse. You do the standard laboratory tests, blood counts, microscopes, cytogenetics, look at the chromosomes, molecular stuff. And we take a sample and we put it into the biobank so that we can do future research onto it, with consent of course. Then those patients would go on, in most cases, to have chemotherapy. So what our plan is also we'll have next generation sequencing, so we know that panel of mutations, all those mutation profiles, so we know what's happening in those particular patient, a particular patient at a particular time. And there's the mutational profile. Remember I showed you all that barcode for an individual person. And that idea then is, you can say, can you map back different mutations with different therapies, even different patterns of mutations with different therapies. So the problems with this is, at the moment, is that mostly patients are treated with standard of care chemotherapy. And that chemotherapy was developed in the 1950s and the 1960s. So we don't really have many therapies that are more recent for this disease. Very few mutations of targeted therapies. 10 of them have got a therapy, and most of these are in clinical trials, so they're not actually standard of care. And patients, as I've shown you, have more than one mutation, so you may need 4 different drugs, but you don't know which 4 drugs are more effective. So what we're doing here in Belfast is to take patient samples, and we're screening them with standard of care therapies and a combination of therapies using our robot. And so basically, you take your patient samples, you put them onto a plate like this, which actually has 384 different wells in there, so you can do a whole patient for a whole screen of different drugs. We're taking a combination of therapies, around about 45, looking at them in single agents, in double agents, in combinations, so we're covering every particular combination of therapies. And here will be the layout of the therapies here, different patients, different drugs, there's only 6 here for simplicity. Looking to see which ones are more effective against the patient's samples at diagnosis. And that will give you a sort of a pattern here, so that you would say, whoops, I've done it again. Here, patient 1 may only have to have drug A. Patient 2 would have B, D and F, 3 C and E, so on and so on. So you have a better idea of what therapies would actually target the disease at any particular time. So, looking at this, we're going to we're bridging the gap between the patient into the laboratory and back to the patient by taking the diagnostic workflow, doing testing both in vitro and in the laboratory, identifying drug candidates, suggesting the appropriate treatments for the patient, monitoring the disease response to make sure that what's changing and what's like that, and do the circle again once patients need it to make sure we're hitting the right targets at the right time with the right therapies. So I called this talk at the beginning a hidden cancer. I want to call it a complex cancer really. It's complex because it has 137 different subtypes. It's complex because it has different number of mutations, from 1 to over 80 genes involved in each particular subtype of blood cancer. Complex outcomes from cure, basically, or almost as cure with the CML, which I showed you, to very poor with the AML and everything in between. And its complex therapies are needed, from single drugs such as anatinib to the complex therapeutic combinations we just talked about for AML. So I'd just like to end by thanking some of the people who are funding some of this research, and thank you for listening. I'm sure you probably have got a sore head after listening to this stuff. Thank you very much.